Welcome back, everyone. And I'm Kwabena. I'm a PhD student at the Research Center ULISH Enabling Technology Group. And today I'll give tidbits of my PhD topic. And for the sake of today, I've titled my talk Syndrome Bus Research Disease Induces Heterogenic Allocation of Carbon in Growing Sugar Beet. So I believe the French word here, syndrome bus research, which I'll be referring to as SBR, is quite new to most of my audience today. And before I dig deeper, I will introduce us to this French word and what it's doing to sugar beet plants. So SBR is a fast-spreading sugar beet disease basically in mid-Europe. And what the disease does is that it reduces absolute sugar content and biomass of affected plants by 25 and 5% respectively. The symptoms of this disease is quite remarkable for both above ground and below ground crop organs. And in above ground, we've observed some proliferation of new leaves and also yellowing of new leaves. But these symptoms become apparent at very later stages of infection. If we dig into the below ground section, we see some brownish discoloration in the vascular bundle. So if we slice into sugar beet of SBR infected plant, we see that portions of the cross section show some brownish discoloration within the vascular bundles. And this is basically the main symptom expressed for sugar beet infected plants. And this disease is caused by two main so-called phloem-restricted plant pathogenic bacteria. And the first one is the proteobacteria Candidatus acinophonos phytopathogenicus and the phytoplasma Candidatus phytoplasma solani. So this disease is a so-called vector-transmitted disease. That means that it requires a carrier to transmit the disease into, to transmit the pathogens into host plants. So what this planthopper here called Pentacillidius leporinus does is that it visits innocent plants, feed on them, and try to introduce the pathogens simultaneously into the phloem sap. So among several intricacies revolving around the subject matter of SBR, the mechanistic understanding regarding crops infected with sugar beet is currently incomplete. And since it's a phloem-restricted plant pathogenic bacteria, the trajectory of the carbon resources within the phloem and the development of the whole beet becomes an interesting niche to study. So to do this, we asked ourselves two questions. The first one, we wanted to understand how this disease alters structural development of sugar beet plants. Again, we wanted to have insight into how SBR disease affects carbon distribution dynamics in basically below ground part of the sugar beets. Now, for sugar beets, the below ground forms the main economic value, so it becomes important for this subject. So to do this, we set up an experiment in such microcosms. We grow sugar beet plants, basically one single genotype with two groups, the inoculated and non-inoculated group, to establish the disease infection. We confine young plants and the plant hoppers, which I've shown on the previous slide, in this kind of tent for an inoculation access period of 11 days. And through this 11 days, we hope that indeed the plant hoppers were hungry, furious, and fed on the plants to introduce the SBR pathogens in them. So before I go inside my results, I would like to give us an overview of the main phenotypic approaches I've employed and how I've achieved and analyzed my phenotypic data. So as I indicated earlier, we are interested in the structural and carbon allocation dynamics of infected plants. And to acquire structural information, we've employed a plant-dedicated MRI platform to non-invasively image basically the below-ground structural development of sugar beet. And for carbon allocation dynamics, we've employed a tracer transport technology and positron emission tomography to monitor or track the fate of carbon within the plant. So the business of the positron emission tomography is quite tricky. What we do here is that we supply a radioactive carbon C11 in the form of 11CO2. And the assumption here is that the plant will take up the 11CO2 and incorporate it into the photoassimilate. And then with the help of our PhenoPet platform, we are able to track the movement or the distribution pattern of the labeled photoassimilate within below ground of sugar beet. So after we acquire such expensive phenotypic data, what we do is to reconstruct those images into several projections, basically either two-dimensional projections or three-dimensional projections. And the next thing is to extract the phenotypic trait we are interested in. So from a single-handed MRI, we are able to extract structural information, basically the taproot, volume, and also quantify cross-sectional anatomical features. I will dig a bit deeper later. And again, another possibility is to co-register the MRI and PET images to have a more or less like a structural and functional perspective of how the tracer is distributed within the sugar beet. From this, the last step is to analyze the quantitative data we achieved from the previous step. So I will show some results on what we've achieved so far. So here I'm showing temporal development of sugar beet. Basically, these are the 2D slices of the sugar beet. So we kind of monitored the development of the cross-sectional features at 3 different spatial levels. So this is what we define as the upper section of the beet, the lower section, and the mid section and the lower section. We wanted to see how the disease affects the plant across different spatial sections of the plant. So here we've observed that in the healthy samples, we saw a clear defined anatomical features, unlike in the disease samples. You might probably appreciate it more if we have a closer look into the upper 2 slices at the last imaging date. So in a control sample, we see a clear defined anatomical structure for a healthy sugar beet. If any sugar beet enthusiast is here, he or she will disagree with me. This is how clear a healthy sugar beet looks like. But on the other hand, in the infected sample, the disease sample, we've seen that there was some distortions in the cambium ring formation, and there's a smearing of the annular rings. And also, if you compare these MRI intensities, we see that sections of the disease showed a brighter intensity than in the normal, healthy, cross-sectional sugar beet. So more on the structural data, we have quantified taproot volume as a direct measure of storage root development. And here what we did was to generate a binary image from the raw data we acquired from the MRI, and then again use a median filter to remove all fine structures like lateral roots, and the remaining orange I'm showing here define the taproot for us. So we first generate a binary image from threshold segmentation, and then we remove all fine root structures by applying a certain median filter and then generate a taproot from it. So here we observed that running about 49 days after inoculation, there was a reduction in beet volume, and this progressed until the last imaging day. Again, the next thing we did was to quantify the cross-sectional anatomical features here. So one unique feature about sugar beet is that in between these rings, the cambium rings that I'm trying to depict here, is where the plant stores most of its sugar. So we try to quantify what happens there in terms of infection. So here we contoured just the 4 inner rings and applied a certain radial analysis to determine the width of the inner ring sections by ignoring or removing the central core. And here we also observed that as early as 42 days after inoculation, the ring sizes, basically the ring width, was reduced by the presence of the SBR pathogen. So The next session of my results, I will dedicate it to the tracer, what we've achieved so far from the tracer distribution dynamics. This was what I think Hendrik was trying to show in his talk, which he skipped, so I'm happy to fill in for you. So here I'm showing a video depicting tracer, the movement of tracer within temporal spatial slices of the sugar beet. So if you agree with me, for the control sample, we observed that the tracer filled most of the cross-sectional part of the sugar beet, but in the infect disease sample, we saw that some parts of the beet was receiving little to no photoassimilate, and these parts were also the part that was showing the bright signals as I've shown previously on the earlier slides. So here we observe a kind of sectorial distribution of the recently phased photoassimilate, in the sense that the part of the beet was receiving little to zero tracer. In fact, we didn't do this just for a snapshot. We traced back and we did this temporarily also. And here we kind of had indication of the sectoriality was already showing up at 42 days after inoculation, but it was really indeed very prominent at the later stages of infection. One thing I forgot to mention earlier on is that in these figures I'm showing, the gray part shows the MRI and the colored section shows the tracer we've applied. So the next thing we did was to estimate heterogeneity in the tracer distribution as an indicator for deviation from normal distribution. So normal distribution as in how homogeneous the tracer was distributed in the healthy sample in relation to what we've seen in the disease sample. So we did this for each sample, beet sample, and every circle I'm showing here represent 10 spatial slices. Like in the video, I was showing a couple of slices, but here we did it for 10 different slices for every sample. And we fit a linear model, and we observed that in the control samples, which I'm showing in blue, dotted lines, we didn't see any strong relationship, but in the disease samples, we found a strong relationship where we observed around 3% increase in heterogeneity per day in our measurements. So let's have a flashback here. Earlier on, I indicated we want to understand how SBR alter structural development of the beet. And indeed, we have seen that the disease is reducing the beet volume and also the inner ring width. And what I haven't shown today is the biomass, but we have a clear indication that the biomass is also reduced. Moreover, we've also observed deformation in the cross-sectional anatomical roots, anatomical features. Again, I wanted to know how the SBR affects carbon distribution dynamics in below ground part of the beet. And here we see that, as I showed in the video, there's a form of sectorial distribution of carbon in the presence of the pathogen. Moreover, we've seen an increase in heterogeneity in the presence of the pathogen. So from our results, we have a detailed catalog of how this disease is affecting sugar beet plants. And what we could do next from this is to use this as a basis for judging susceptibility and also selecting resistant genotypes to benefit us for future breeding programs. Again, it will be beneficial for us to also determine the shoot physiological responses during SBR sugar beet interaction, and this will provide us a holistic overview of structure and functional relationship existing between above ground and below ground parts of the beet. And one thing I would also want to emphasize is that the SBR disease is also extending its host range in mid-Europe. That means it's affecting other plants, and it will be beneficial for us to apply our systems into other pathosystems like in potatoes. I'm very thankful to the people I've listed here from the Forschungszentrum Jülich, in Germany and also the Institute for Sugar Beet Research in Germany. And thank you for your attention.
Thank you. We have time for a question. Well, okay.
Thank you for a very nice presentation. So I'm super happy to see the progress. Yeah. So just a quick question for you would be, when you do MRI scanning, right? You have a different time point scanning. So have you noticed any sort of effect on the pathogen side?
Of course, we are measuring sugar beet switches. So with MRI, we are basically measuring protons. That's the water in the samples, and the water content in sugar beet keeps increasing with time. So what we did was to tune and match our system to suit every stage of measurement. So of course, we have some little minute signal differences, but it doesn't really manifest in a negative way. Yeah.
Thank you.
I was wondering, have you also measured the starch concentration in the sugar beets, either by MRI or directly in a chemical way?
Of course, we could do this by, for example, chemical shift imaging, but we haven't established protocols for this particular niche of study. Yeah, but so it's a no. I haven't done that.
I expect the starch concentration has decreased, right?
I cannot conclude on this because if I think about the density of the infected and the control plants, I'm not assuming a lot of differences, but I will not say no or yes for now because I don't have a data. But if you ask me, I will say I don't assume a lot of differences in terms of it's not starch but sugar content at this stage, because we screen at the very infant stages of the infection. Maybe at later stages we will see more, but at this we've done our analysis until just 63 days after inoculation, and at this stage I'm not assuming more.
Yeah, over here. This carbon 11 stuff that you are working with, can you describe a bit how complicated it is to work with this material?
It's pretty complicated. So in Germany, we call something challenge shots. You need some radiation protection stuff to even, it's a full controlled area. So what you don't want to do, we produce, I think for each measurement, we produce 50 megabecquerel of the radioactive CO2, but we cannot be that exact. So we first trap it in a sieve and then put it in our system, and because it has a very fast half-life, it has a half-life of 20.4 minutes. We are able to do some algorithms to just admit just 50 megabecquerel in our system. But during this time, of course, we are not allowed to go there. We just keep the plants. Or our setup, I think I showed the literature. If you are really interested in this, you can have a look in our setup. It's in a climate controlled area, so everything is under control. It's quite dangerous, but not that dangerous.
Do you think in UNIF we will continue working with carbon 11 in the upcoming 5 to 10 years?
First of all, I'm not a policymaker, but as a beneficiary from this, I will say yes, because we are not just using it for plants. There are I think Neuroscience Institute also in Ulysses has advanced system, and they are really deep into application of C11 in their systems. Yeah.
Okay. Yeah. Thanks.